This is a B4 bioenergetics video looking at photosynthesis, in particular factors that affect the rate of photosynthesis. Now, introducing those factors that affect the rate of photosynthesis, we call these limiting factors. Okay, we've got light, carbon dioxide, temperature and chlorophyll. A limiting factor is one of the factors is restricted, the rate of photosynthesis will be below the maximum possible rate. And we talk about getting things at the optimum conditions, the perfect conditions, the best conditions. Now, if we've got our light intensity at the optimum condition, then something else is a limiting factor. Once we've got our optimum conditions for, say, light and carbon dioxide, temperature could well be the limiting factor. So it's about trying to create the optimum conditions across all of these conditions to get the maximum rate of photosynthesis. And we have to think like a farmer, okay? Thinking about profit, thinking about high yields to get the best from our plants. Now, you probably remember seeing this program used to model the rate of photosynthesis. And what we did is we changed the distance of the light. Rather than looking at the distance that the lamp was, which they may ask you in an exam situation, we used this light meter in the middle here, and this was able to tell us different light intensities. So we created a graph that had light intensity at the bottom, and then size of oxygen bubble, which we could see just here, and then we enlarged it to read it better here. We could see the size of the oxygen bubble. Now, the more oxygen produced, the greater the rate of photosynthesis. So we could look at distance of the lamp. As the lamp gets further away, there'll be less light intensity and there'll be less photosynthesis, therefore less oxygen produced. But it was nicer because our graphs are usually light intensity on the bottom. It was better to use the light intensity meter in the middle. Okay, we call that a light meter, measured in lux, L-U-X. And we looked at how much oxygen, how big was that oxygen bubble in 10 minutes. Now you could, if you had this set up and because obviously you've got the pond water in water, the pond weed in water, you could count those bubbles coming off. Okay, so if we set this up real life in the lab, we could, for say 30 seconds or a minute, count how many bubbles of oxygen are produced. Okay, so we could have a clicker and click every time we see a bubble, okay, for 30 seconds or a minute, and that could be our way of measuring the rate of photosynthesis. And there are other programs. If you go on Reading University photosynthesis experiment, they have a setup similar to this where you can change the distance of the light and you can actually click the bubbles as they're produced, and there's a clock on there as well. So it could be the length of the oxygen bubble produced in a capillary tube, which is what you can see in front of you, or it could be counting the number of bubbles produced in a particular amount of time. Either way, both of them are over a set period of time. And as I said before, we're using the volume of oxygen produced as our indicator of rate of photosynthesis. So let's have a look at some of the results. You see at the bottom there it says the easiest way to monitor the rate of photosynthesis is to measure the amount of oxygen produced in a given time. This can be done by investigating using pondweed. Now, we could, just like when we talk in C6, rates of reaction, we can talk about, okay, well, let's do a fixed period of time, how much gas do we get? Or we could do, let's time to see how long it takes to produce, say, 20 bubbles. So there's two ways of looking at it. I think the best for this is fixed period of time and how much oxygen is produced in that period of time. Now we can see here the first graph on the left is light intensity. The orange bar represents where light intensity is a limiting factor. Once we reach the optimum point where we've got the best rate of photosynthesis, it would be silly increasing the light intensity. As a farmer, that would cost more money to increase the light intensity and for no profit, because the rate of photosynthesis does not increase. So that's why it's useful for a farmer, okay? It's all about profit, it's all about high yield, Let's find the optimum and then let's move on to the next limiting factor. So we can see light intensity there is a limiting factor at first. Once we reach the optimum, something else has become a limiting factor. Now sometimes on questions they give you a section of blank paper as well as a section of lined paper, and this is where they're asking you to draw these kind of graphs. Okay, they're asking you to put a dash on there. Where's the optimum? Where would you find that? Okay, so let's look at the writing to go with the light graph. Light is a limiting factor, affects the rate of photosynthesis up to the optimum. After the optimum, there is no benefit increasing the light and it costs more. Let's look at the second graph. We're looking at carbon dioxide, same shape of graph. We can increase the carbon dioxide. This is going to be in a greenhouse situation. Okay, it's not going to be in a field because the carbon dioxide is going to just spread out diffuse through the air. Okay, so this is in a greenhouse situation. If we increase the carbon dioxide, and we could do this for the pondweed as well, you can add carbonates in that increase the carbon dioxide in the water. We could do that on the program. Again, once we've reached the optimum condition, something else has become a limiting factor. And there's no point pumping in any more carbon dioxide because it will make no more difference, but it still costs money. Temperature. Okay, this is again linked to enzymes. Okay, so we've seen this graph time after time in terms of temperature, looking at how it affects enzymes. And when temperature is the limiting factor, it can be if the temperature is too low. And we need to think about, again, rates of reaction, C6. Particles don't have much energy, so therefore they don't collide very often. Okay, there's less frequent collisions. And then above that optimum, where the temperature gets too hot, we've got wilting of the plant because it's got so hot that lots of water is evaporating from it. And eventually we've got the denaturing of enzymes. Okay, so limiting factor in two parts of the graph there, optimum around that 40 degrees C. Chlorophyll is the final limiting factor that they just brought into the course. Okay, and it makes sense. We talked about chlorophyll being like the oven. Okay, the more ovens you've got, the more cakes you can bake. Okay, the more chlorophyll there is, the more photosynthesis that can occur. Okay, and limiting factors are important in the economics of enhancing conditions in greenhouses to gain the maximum rate of photosynthesis while still maintaining profit. Let's not waste money creating conditions that are beyond the optimum and can't make a difference to those plants. Hope you found that useful. Thank you.